Cool, we're good.
All right. Hello, welcome everyone to the first IWGC webinar. Today's speaker is going to be Caio Brunharo at Penn State. Before he begins, I just wanted to point out the tentative schedule of speakers for this webinar series that will continue throughout the rest of this year. We'll have approximately one speaker a month. So take a look at that speaker list and pick out the talks that you want to see or come to them all. For those of you who don't know, I'm Eric Patterson. I'm at Michigan State University. I'm on the board for the International Weed Genomics Consortium. And with that, I just wanted to, introduce the seminar series and say I'm so excited that we're getting this started off. And we hope it's going to be really great. If you have any questions, please don't hesitate to put them in the chat and we'll address them as quickly as we can. And the other note is that this will be recorded and distributed on the IWGC website, which is weedgenomics.org. So please, once we get this recorded and uploaded, check there if you want to view it later. Anything else, Caio?
I think that's pretty much it. Oh, and about the times, so the idea is to have a seminar in tentatively the last Monday of the month. Sometimes it's not gonna work Monday, so we're gonna move to a Tuesday or Wednesday at a time that we're gonna decide later. So it's still working out the details, but we're going to advertise whenever we have a set date.
Absolutely. All right. Well, thank you all for attending today. I'm looking forward to it. Caio, why don't you take it away?
All right. Thanks, Eric, for that introduction. And I'm super happy to be here today sharing with you some of the work that my lab has been doing over the last, I'd say, 3-4 years on herbicide resistance in Italian ryegrass or Lolium multiflorum. So you're going to probably see me switching back and forth between Lolium multiflorum and Italian ryegrass. But what I will be sharing today is some of the genomics work that we did looking at herbicide resistance, specifically to non-selective herbicides, glufosinate, glyphosate, and paraquat. So, this is all, the answer is very weed centered, so I'm not going to spend too much time here, but weeds are super important in agriculture. They have a big potential to reduce losses. Usually we do a good job overall controlling them, but that costs a lot of money as well. We spend a lot of money controlling weeds. We lose a lot of money because they take away the resources, and not only direct costs with weeds, but also they can have some indirect costs like reducing marketability of the products. Let's say you have a seed lot that you want to sell, and then it's contaminated with weeds, so that seed lot might lose some of the value. It can drive a loss of land value as well. Can be alternative hosts for other pests. And we mostly control weeds with herbicides. They are the #1 way that we control, and that's a problem because we over rely on herbicides, leading to herbicide resistance. And this is going to be a key topic in my presentation today, is the evolution of herbicide resistance. So far, we have selected as of this morning, 530 herbicide-resistant cases of herbicide resistance, with the United States leading this ranking, followed by Europe as a whole, Australia as well, and then Canada, China, and Brazil. And the problem with herbicide resistance is that they make weed management much harder. In some situations where you have multiple herbicide resistance, farmers really end up running out of options sometimes. And this is a very interesting number, that we find herbicide resistance to 21 out of the 31 mode of actions that we have registered nowadays, which is very interesting because that tells us that weeds can find a way of evolving to multiple different herbicides. So how does herbicide resistance happen? So herbicide resistance is typically classified as target site and non-target site resistance. Target sites are the types of resistance where, as the name implies, target site is modified. So you can have mutations where that lock-in key mechanism between the herbicide and the enzyme that's targeted, it no longer works. Some sort of mutation or deletion can happen. And also another way that we classify another type of target site herbicide resistance is when we have increase in the expression of the target site gene. So this is an example here of work by Todd Gaines, where a susceptible individual could have only one copy of the EPSPS, which is the target site of glyphosate. And then because of evolution, the resistant plant, instead of having one copy, can have many, many copies of that EPSPS target site. So there's no enough glyphosate inside the cell to inhibit all the enzyme, and the plant can continue with its regular functions. As opposed to non-target site. So non-target site is a little bit more complicated because, when we are dealing with target site resistance, usually we know what the target site is for a herbicide, but non-target sites, we don't have that. So many things can happen. There can be physiologically, there can be an exclusion of the herbicide from the cell. There can be enhanced vacuolar sequestration to the vacuole or some other organelle excluding or keeping the herbicide away from the target site. There can be also herbicide metabolism as well, where the herbicide can be broken down into less lethal forms. And this is a more recent type of mechanism of resistance, which is the rapid necrosis. But as you can imagine, there can be many options here, many ways that the weed can evolve resistance with different enzymes being responsible, different types of modifications into the gene or outside of the gene. So it's a little bit more complicated to characterize that type of resistance. So there are many, from that slide, the take-home message is that there are many questions that are left to uncover. We know very little still about non-target site resistance. So how does herbicide resistance evolve, specifically non-target site resistance? Does it come because of new mutations in the genes or outside of the genes, because of gene flow from standing genetic variation that is already in a population? What are the genes that are controlling non-target site resistance? We know a lot of the physiology, but we still don't know a lot about genetics of non-target site resistance. And there is the idea that perhaps one master switch is controlling target site, non-target site resistance to more than one mode of action herbicide, or perhaps multiple genes are necessary to confer resistance to multiple herbicides. What's the genetic architecture of non-target site resistance? Is there just one locus or many loci? What's the effect size? Is it just one gene that is responsible for a large effect in giving resistance? Or there are multiple small loci genes involved, which is the concept of oligo or polygenic and monogenic resistance. And also the patterns of parallel and convergent evolution. How many ways and many times can that type of herbicide resistance evolve in one location or in multiple locations individually and independently? And that's where omics tools can be handy, because they can help us answer a lot of these more kind of traditionally challenging questions. They can help us in many ways that I'm gonna be showing you in this webinar. So why multiple Lolium multiflora, Italian ryegrass? It has been the focus of my research for the last probably like 10 years. I did my PhD with this species and I continue working on this species. This is a weed that is important in many cropping systems, not only in the United States, but around the world. We have a few examples, photos that we took along the years. This is an example of a paraquat resistant population in a prune orchard where this photo was taken a few days, probably like 4 days after a paraquat application. So we see very minimal necrosis from the herbicide. This is a Italian ryegrass plant from a wheat field. And then this is a lot of Italian ryegrass that survived to an application of a group 1 or ACCase inhibitor in clover in Oregon. So it's important in many different cropping systems. And lowly multiflorum is particularly interesting to me because we have reports of resistance to over 70 populations around the world. And just for comparison, we hear a lot about other species as well, like Amaranthus palmeri or Palmer amaranth, water hemp, or Amaranthus tuberculatus, and Coniza canadensis or horseweed. And those numbers are very high as well, and for those species, which is no different for Italian ryegrass. And a closely related species to Italian ryegrass, which is Lolium rigidum, if we combine those two species, then we have about over 120 reports of herbicide resistance. So it's a genus that evolved resistance multiple times and has the potential to be very diverse in terms of resistance evolution. I've worked with populations of Italian ryegrass with resistance to more than to 4 modes of action. So we can see this species has flexibility to evolve resistance to many different modes of action. And I'm particularly interested in these non-selective herbicides because glyphosate, and paraquat are super important for these cropping systems where I have worked on in the past and still work on. And growers rely a lot on these non-selective herbicides for Lolium multiflorum control. Around the world, we see most of the populations were identified in the United States, but we can see some other countries like Brazil, Argentina, Chile, and some countries in Europe as well, where resistant populations were also identified. So the objective of this particular project I'm showing you today is the first one was to generate genomic resources for Italian ryegrass. When we started this work about 2.5 years, 2022 more or less, so over two years ago, we didn't have a reference genome for Italian ryegrass, so a lot of our questions were really hard to answer without this resource. So the first one was to put it together and then start dissecting the genetic architecture and asking questions about resistance evolution to those non-selective herbicides. And ultimately, something that we really wanted to know is to identify the candidate genes for functional characterization. And this is something that's currently ongoing in my lab. And why we want to identify candidate genes and understand genetic architecture, because this can be give us good information regarding adaptation to human-mediated stresses. I think that weeds are a really valuable source of information on how plants respond to adverse conditions, not only to herbicides, but weeds, if you think about it, weeds are constantly under stress. They are under stress because of our management practices, not only herbicides, but other things, cultivation and mowing and things like that, but also because of competition with the crop, because environmental stresses. So if we can harness that information that weeds can give us, then we can perhaps breed more resilient crops that can tolerate to other things like environmental stresses, competition, and so on. And just how biology can find ways to evolve under selection pressure. And some of what my lab really focuses a lot on is to develop solutions. We are really interested in using that information to develop solutions for farmers, such as quick assays for resistance detection, so that farmers can have a better informed decisions on what to spray or what to do about a population that they have in the field. So why do we need a reference genome? And I think that's really a good slide that I wanted to show you with this audience in particular, why we need a reference genome. Because the reference genome is a representation of a genome of our species. And now we are actually moving into a new era, which is the era of the pangenome, where we don't have only a single plant that represents the entire species, but also multiple ones. And the lack of a reference genome can pose us really can be very important, limit us on how we the questions that we can ask. And that's because a trait, if you think about herbicide resistance, in my case, a trait can be influenced by many genomic features, like changes in structural variation in the genome, like duplications, deletions, inversions. And this is something that is shown here by a work by Chris Benson working on POA species. It's a tetraploid. So they found that depending on the genotype, some of the genotypes have changes in the genome, in the chromosome size, and things like that. So there can be a lot of variation in the genome that can also reflect on the phenotype. So you see a couple of phenotypes here, different growth habits of this POA annua species, and that could be related to some of this structural variation in the genome. Also understanding mobile elements, no Condy regulatory elements, and just single nucleotide polymorphisms or SNPs. They could be associated with some of the traits that we are interested in. My particular case, herbicide resistance. So how we can assemble a reference genome? So the first thing is to identify an individual plant that represents your species. It doesn't need to be necessarily a good representation of your species if your goal is not that. So for example, for my case, I decided to choose a herbicide susceptible plant that we characterized in the past, because for my goals, having a susceptible reference genome made sense. So that's what I went for. Also, it's important to get really good quality DNA and RNA, because those two will be important when assembling and annotating the genome. Nowadays, long read is preferred. So you want to sequence, have a good quality DNA so they are not fragmented, and then you can sequence DNA and RNA, assemble computationally all the reads. You can get some extra type of data, which is high-C data. You can look it up later, but basically we're taking advantage of the 3D structure of the DNA, and we assume that closely connected stretches of DNA, they are also closely when they are sequenced. We also generate RNA information because we want to know where each gene is expressed. So that's really important. Another thing I should point out here is that you want to extract RNA from multiple tissues, because not all express genes, not all genes are expressed in the same type of tissue. There can be differences. And also identify other genetic features. You can take advantage of databases that are already put together, open source, and they can help you annotate your genome. So specifically for Lolium multiflorum here, we generated PacBio Hi-Fi, which is a type of just a type of data where there is a very high confidence in how in the DNA sequence, so very low error associated with the sequencing. We use Hi-C data, and we implemented 3 different software to put together or assemble the genome. And then for the annotation of Lolium multiflorum, we use Hi-Fi data. So we extracted RNA from leaf tissue, roots, pistil, and stigma. And again, that goes back to understanding where each gene is expressed depending on the tissue. And then we use a series of software to put it all together and annotate the genome. Basically, annotation just means where each genomic feature is located in the genome. So we got a really good reference genome put together, we obtained a genome size of about 2.8 gigabases, and we had 253 contigs. That really tells us this number is really important because that says over 90% of the genome is located in 7 contigs. So that really makes sense because Lolium multiflorum has 7 chromosomes. Most of the DNA that we found was repetitive elements, over 80%. So that means they're not necessarily genes that we would have any function directly. They are repetitive elements. Annotated genes, we have about 50,000, and Busco analysis, this number just means how complete your genome is in terms of genes. So it's a pretty high number here in terms of Busco, and it aligned well with flow cytometry data. So flow cytometry is a different technique that quantifies how big the genome is. So there's a really good alignment, over 95% in terms of equity here. So we have a reference genome. Now what we can do. So after you have a reference genome, I think it's really important that you develop a solid experimental design and focusing on your hypothesis, because you're generating some data that's not cheap. So I think the first thing that you need to do is to really develop a solid experimental design. You might have tons and tons of money, but if your experimental design is not well adequate to answer your questions, it's not going to happen. It's not going to work and magically give you answers. So that's the first thing I really recommend people doing. And then decide what type of data will be able to answer your questions. Like, you don't want to, for example, do a QTL analysis if you are interested in understanding the gene expression of a particular gene or your entire transcriptome. So really, understanding what the questions are. And then you can start generating the data. There are multiple types of data that you can generate that I will be sharing with you in the next few slides. So now moving on to our specific non-selective herbicides, I'll start with paraquat. So paraquat is a herbicide that, again, is non-selective. It's being reported with resistance in 75 times in 33 species and 20 countries. And we don't really know what are the resistance mechanisms. People have done research in Arabidopsis mutants, and this has given us some good information, some good leads on what could be happening. And particularly in weeds, it seems to be related to exclusion. So paraquat, the target site of paraquat is located in the chloroplast. And our previous data suggests that paraquat is not getting to the chloroplast. So there's some sort of excluding mechanism happening. What I find really interesting is that when we look at the level of resistance, weeds have a much higher level of resistance compared to what those Arabidopsis mutants are surviving. So that could indicate that Arabidopsis might not be completely directly translated to weed species, particularly Lolium multiflorum. So we want to understand paraquat resistance here in this project. And again, I bring this picture a little bit bigger now, showing you that we don't see much symptoms in these particular populations that we found in California. And our data suggested that likely a paraquat is being transferred or pumped or sequestered to the vacuole of the weeds, but we still don't know what are the genes that are doing that type of transport. So our experimental design, what we did, we started by doing, we decided to do a QTL analysis or a quantitative trait loci analysis. This starts with you crossing a resistant individual parent with a susceptible, and then crossing that filial generation. And the idea is to artificially recombine the susceptible and the chunks of the genome throughout the chunks of DNA throughout the genome, and then looking at the phenotype, seeing if they are resistant or susceptible. So that's the idea of an F2 mapping population. So phenotyping here is very crucial, so you can identify which populations are resistant to susceptible. Because we have a high level of resistance, it's pretty straightforward. Just spray and see what dies, what doesn't die. And then after that, you need to associate the phenotype with the genotype. So in our case, we did a type of reduced representation sequencing. It's called NextRed. But basically, the idea here is to take the DNA that is in a long stretch of DNA, and then we prepare our samples by fragmenting it somehow. There are many different types of assays that we can do for a red-seq. In our case, we use the NextRed, but basically the idea is to fragment the DNA and then sequence the ends of the smaller fragments. And then with that information, you are going to map those reads to the reference genome, and then use in silico or using a computer to identify markers throughout the genome. In our case, we use single nucleotide polymorphisms, and then use other statistical analysis to identify the associations between the phenotype and the genotype. So that was the QTL analysis. Now, like I said, the QTL, you are artificially shuffling the genomic regions of your samples. In the GWAS, the genome-wide association study, is a little bit different in that we, instead of artificially shuffling the DNA, we are taking advantage of natural shuffling that already happened over the years, and then sequencing those individuals after you phenotype them. So in this case, we took 3 paraquat resistant populations and 3 susceptible populations, and then phenotyped them and sequenced. For the sequencing here, we didn't do a reduced representation. We actually did a shotgun sequencing or resequencing of 95 individuals, susceptible and resistant. There are a few advantages of doing resequencing versus the reduced representation. Because reduced representation, you end up only sequencing about 1% of genome, versus here, you sequence the entire genome 10 times, and then you align that to the reference genome, and then you identify the markers. And finally, for the paraquat resistant study, we also used a few population genomic approaches, namely genome-wide FST analysis, which is a measure of divergence between the resistant and susceptible populations. And then the higher that FST number, that means there's probably a selection pressure happening because of paraquat resistance. We also looked at genetic diversity between the resistant and susceptible individuals, with the assumption that low diversity regions could indicate selection. And also another measure called extended haplotype homozygosity that has to do with how the resistant selection pressure from paraquat will change the genetic landscape around the resistant alleles. Okay, so moving on to some of the results. For the QTL analysis, we identified a couple of regions of chromosome 5. So basically, this plot is showing the genomic locations throughout the genome. Each panel is 1 chromosome, so we have from 1 to 7. And then the delta SNP index is just a number that tells us how associated that marker was with our phenotype, in this case, paraquat resistance. So when this the black line goes above the red line, that means a significant. So we see that significant genomic region around in the middle of chromosome 5 and the end of chromosome 5. And again, this is from the F2 mapping population. This one is the GWAS results, where we see whenever our data points go. So let me go back a little bit. So each color represents a different chromosome, and the y-axis represents the p-value associated with that marker. In our case, we're looking at SNPs, and we see the chromosome 5, a number of SNPs that go above the threshold line, the FDR, the false discovery rate line. So they are likely associated with our phenotype, paraquat resistance. And the interesting thing here is that these 2 regions, they align really well And they are actually two separate data sets, right? So one was from a artificially generated F2 mapping population. The other one's taking advantage of natural recombination happening in the population. So there is a really good, a strong indication that these regions potentially contain the herbicide-resistant variants. When we go over the population genomics types of analysis, here I'm showing you for each chromosome, we have all these SNPs that were compared between resistant and susceptible individuals. And the larger the number, most likely associated with paraquat resistance. So we see again in chromosome 5, there is a strong association with the paraquat resistant population. So from the previous figure, now zooming in a little bit here, we just zoom in looking at those two regions that we found also in the GWAS. These green hexagons are showing, so again, this is a separate analysis. This is a third analysis looking at population genomics. Now I'm showing you FST analysis, where the green hexagons or data points are these SNPs that overlap with the GWAS. So again, some more overlap between the three different analyses. Here I'm showing on D and E, two different regions, again in chromosome 5. And here I'm showing that each row is a haplotype from the resistant or susceptible individuals from the different regions, this region D and the first region that's representing D and second region representing E. And each column represents the variants that were identified. And the yellow box represents the reference allele and the red box represents the alternate allele. So overall, in this entire data set, we observed a haplotype diversity that was 45% lower in the resistant individuals. Again, that has to do with the herbicide-resistant alleles involved. And then finally, this last type of data that we obtained, this is called extended haplotype homozygosity test. Again, the idea is that the larger this chunk of DNA is more associated with herbicide resistance because there's a tendency of those loci involved in resistance to also bring genetic material around it that follows the resistance phenotype because of recurrent selection pressure. Okay, so then what we did next? So we found a lot of stuff here, very strong indication that there is something going on in region 5 of the chromosome 5. So we took those regions that we found associated with paraquat resistance, and then we extracted the genes that are located within 2 megabases from the peaks. And that can be done because we annotated also the genome. So basically we found, we identify each gene that is located and we know their locations because we did that computationally. So what we found there in those 2 megabase region, we found a couple of genes that are associated with membrane transport. One is called NPF 5.10, and that's a nitrate or an oligopeptide transporter localized to cellular membranes. And also this is important because this has been shown to be involved in polyamine transport. So paraquat is a type of polyamine, so that makes a lot of sense. And I did not show you, but we also did RNA-seq and a network analysis, and also show the results from those 2 separate analyses also show that this gene to be important. So there's a really good lead here to be involved in paraquat resistance. And DTX10 is another important gene that is from the MATE family associated with multi-drug resistance. And DTX, which is one of the members of the DTX family, has been shown to give paraquat resistance in Arabidopsis mutant. So that's another one very good lead that could be involved in paraquat resistance. All right, so moving on from paraquat resistance, we identify some candidate genes. Now I want to move on to glyphosate resistance. So glyphosate resistance is important in ryegrass because it's a really important tool that farmers use to control this species throughout the year. Pretty much, there are farmers in separate types of regions in the U.S. controlling them in the summer, in the winter, in different conditions, different cropping systems. But glyphosate is the herbicide that people use for ryegrass control. And we identify, this map is showing where glyphosate-resistant Italian ryegrass has been identified in the U.S. So we see at least 13 states in the U.S. Found herbicide resistance. So that's really important because it starts we can start asking questions in terms of independent evolution, with repeated evolution. Do the populations somehow, are they related, or because because of agricultural activities, we are moving the genes around, or they evolve independently? So there are many questions that we can start asking about that. And there are multiple resistance mechanisms that have been found in Italian ryegrass, such as SNPs associated with glyphosate resistance, position 106 in EPSPS. There's been copy number variation found, not only in a couple of states that I can remember. And from the populations that I have studied in the past, most of the cases are related to non-target site resistance. Okay, so how we did that, this study, to understand or identify or help elucidate resistance mechanism to glyphosate in ryegrass. So we also did a QTL analysis, so I'm not going to go over too much details here, but we use a population that was previously characterized. It doesn't have any target site mutation. We did not find any other non-mechanism of resistance. So we chose that one because that could give us new information on resistance evolution to glyphosate. We also implemented the next red again, the technique that samples the genome and you only sequence about 1% of the genome, align that to the reference genome, and we use software to identify or associate the genotype with the phenotype. So what we found here, so again, on each panel represents one different chromosome. In ryegrass, we have 7. The x-axis are the genomic locations, and the y-axis is the p-value. Whenever the black line crosses the red line, that means a significant association. So we find a significant association on chromosome 4 related to glyphosate resistance. So what we did was to extract the genes that were annotated in that region, and we found about 800 genes. So that's not ideal, right? Because we have 800 genes. What is it? Like we don't know. It's really hard to test all 841 genes. So we're still working on this project. But however, there are a few good leads. So we found tonoplast associated membrane that are involved in ATPase transporters. So basically transporter types of enzymes that could be involved in glyphosate resistance. So again, this is an ongoing project. We have many collaborators who are providing their populations for us to test. But the next step that we are doing, again, 800 genes is not really easy. So we have to find map the genes. So basically narrow down from that 58 megabases region of chromosome 4. Now we want to go even finer. We want to just a few megabases, so then we can start doing functional validation, basically taking that gene potentially associated with resistance, and then we move it to other species like Arabidopsis or rice or something, and then see if resistance still can happen after that making that new transformed line. Other questions that we are also asking currently is about QTL with different populations. We want to understand if they all have the same resistance mechanism or they have different resistance mechanisms. And also develop markers. So we just got some funding for developing markers for quick detection of glyphosate resistance. So we're super excited about this type of work. And finally, the last herbicide I wanted to talk about is glufosinate. So again, glufosinate is a super important herbicide. It's key, especially in the west side, west coast of the United States. Farmers use glufosinate a lot for Italian ryegrass management. So far, there are 10 populations from 7 species resistant to glufosinate. 8 of them are grasses and 2 are broadleaf weeds. And this is unpublished data that we got in last year or so. But we have multiple glufosinate resistant populations of Italian ryegrass that show different levels of glufosinate resistance. The most resistant of them can tolerate more than 10x the label rate. So it's pretty interesting. I've done some work characterizing the physiology of glufosinate resistance in the past. And our previous results indicate that there are multiple resistance mechanisms. So we studied 3 populations total. One was SECO. One of the resistant one had enhanced herbicide metabolism, and the other one we didn't find enhanced metabolism. So we still don't know what's going on in that particular population. So for understanding glufosinate resistance specifically for this project, we did again a QTL analysis where we generated an F2 mapping population. We phenotyped with a little dose of glufosinate. Same thing as I did before in paraquat and glufosinate. Discovered SNPs throughout the genome, and then implemented this QTL SPIC-R R package for identifying association between glufosinate resistance and the genotype. We implemented the FST analysis between resistant and susceptible populations, and we also did a RNA-seq with non-treated plants between that were generated with the F2 mapping population. Similar type of data that I showed before. We have each panel represents one chromosome. Whenever the black line crosses the red line, that means a significant association between glufosinate resistance and the genotype. And we see something going on chromosome 3, likely associated with glufosinate resistance. We also did a FST analysis. Again, FST analysis is a divergence type of measure, comparing resistant and susceptible populations. The larger the number, more likely it's associated with the phenotype that we are working on. So really good overlap between the 2 different analyses. Similar as before, we extracted the genes associated or present around the FST peak, and we found about 150 genes. Some of them were involved in herbicide metabolism and ROS detoxification. So could be associated with glufosinate resistance. We're still working on it. But again, I should point out that this region is 58 megabase wide. So it's a big region of the genome. So still a fine mapping here could help us narrow down what genes are involved in the resistance. We also did RNA-seq, like I mentioned before. We identified 21 genes differentially expressed, and this number is relatively low actually, and that has to do with the fact that we sequenced the F2 mapping population, so that helps reduce the background genetic or gene expression here. And one of the genes that was interesting is within the QTL peak that we identified having 3-fold greater expression. So we're still, again, working on it, trying to working on a fine mapping population. All right, so I'm just running out of time here. So just for conclusions, the overall conclusion that I would like to share with you is that, again, we looked at non-selective herbicides, those herbicides that pose a lot of selection pressure on weed populations. They have a very high kill rate. And what we found is that few genetic variants have large effect size on herbicide resistance. And that's because we found a single genomic location for each of these genes on chromosome 4, 5, and 3, 4, and 5. So that tells us that one thing it tells us is that it's important, large effect size is important, so a few genetic variants involved in herbicide resistance. Another thing it tells us is that they don't have the same resistance mechanisms. They evolved separately. They are not related. So we haven't looked at this, but likely they are inherited in different fashions. So you can separate resistance, which can be very important for this primarily outcrossing weed species. And also support the idea that evolution to rapidly changing environments require a few loci with large effect size. So just to summarize, the glufosinate, we did a QTL-seq analysis, FST genome scans, and RNA-seq to find a couple of very interesting genes potentially involved with resistance. They have herbicide metabolism and xenobiotic transport across membranes. For glufosinate, we did a QTL analysis only, and we found a region of chromosome 4 involved in resistance. And in paraquat, we did a number of analyses, QTL-seq, GWAS, genome scans, RNA-seq, and network analysis to find a couple of very interesting genes could be involved in paraquat movement in the cell. And we're still working on it, right? So, and the more, I feel like the more we generate data, more questions we have. So for instance, for glyphosate, we are still trying to understand whether the different populations that were found in the US, if they are the result of repeated evolution or just the result of agricultural activities where we are moving genes around because we are transporting seeds, we're transporting people, and so on. So how does glyphosate resistance evolve in ryegrass? We're still fine mapping, generating mapping populations on for the different populations to understand the genetic architecture, and also developing molecular markers. Hopefully they can be implemented for understanding or for helping farmers to decide on management practices. And glufosinate resistance is another work in progress. We are generating mapping populations to find map resistance, and we intend to do functional validation of the genes, not only functional validation of glufosinate, but also for glyphosate resistance as well. Okay, so in terms of my thoughts on omics, I see the list of folks that I recognize the names here, a lot of students in the audience. So I want to share with you, and my opinion is, and I'm not, usually I'm not right, I'm always wrong, but something that you should consider very well, carefully, is experimental design. Because, it happened to me, sometimes I spend so much money, resources, and money is not only cash, like it's time, like time is money. So you spend so much time and effort and energy putting on a project to later realize that your experimental design wasn't ideal and didn't answer the questions that you wanted to answer. So I think this is really important. Understand the biology of your weed. Weeds are very different, right? So ryegrass is an obligate outcrossing species. Some species are self-pollinated. They have different types of amount of genetic diversity and so on. So that's important to understand what the makeup of biology of your weed is. Good phenotyping, also super important, understanding whether they are not only resistant. Resistance is usually straightforward, but not always, right? Collaborate, don't be afraid of reaching out to people. There are a lot of people out there interested in collaborating and helping out, especially students. So don't be shy, just reach out. Understand the limit of each technology and what it can do for you. And also stay on top of current technology, because things are moving super fast, getting cheaper and more accessible as well. So I think it's important to be on top of things. With that, that's my last slide. I just wanted to thank a lot of the collaborators that I had along the years, putting, getting this data together, analyzing everything and writing papers. So Adrian especially is my current PhD student. He's working on a lot of the crossings to understand glyphosate and glufosinate resistance. So we are gonna probably hear from us in the near future with some more information on glufosinate and glyphosate resistance genes. Thank you so much. And I'm happy to answer any questions.
You do have 3 questions lined up in the chat. I can read them out to you if you want to just answer them.
Sure, yeah.
First question is, how big were the F2 populations? Were they from the same F1 cross?
So the F2 population, okay, so how big they were big. Like I had like 300 plants, but I only wanted to sequence, I only had funding to sequence 95 individuals, and I wanted to be half and half. So out of the like 300, I picked 49 or 48 resistant and 48 susceptible, and they were the result of the F1 cross.
Next question is, why did you use both GWAS and QTL analysis? Do you find that one is easier or more robust than the other?
That's a good question. So I did both because I wanted to have orthogonal validation, right? So I wanted independent analysis, and hopefully they reach the same conclusion. So that's why I wanted the QTL was a separate one where you identify, you make an artificial population, you sequence that, you phenotype and sequence, versus a GWAS where you take advantage of natural variation happening in the population. So the benefit of the F2 or the GWAS is that you have the potential to identify the actual genes. And that's also true for the F2, but for the F2 analysis, your genomic blocks will be much larger because you don't have as many, you have like one recombination for each generation, right? So you don't break those associations, those linkages between genes very often. You would need a very large population. So by sequencing like just 100 F2 populations, your genomic regions would still be big. Versus GWAS, you can narrow down even further. Although it's more expensive to do GWAS than a red-seq or QTL analysis. For a ryegrass, right? It changes so much between the different organisms because if you're working on, Arabidopsis that the genome is 100 and something megabases, then it's not perhaps too challenging to do a GWAS versus ryegrass that has almost 3 gigabases genome. It's much harder, it's more expensive to do a resequencing type of data generation.
Next question from Luan. Great work, Kaya. How do you intend to make molecular markers to diagnose metabolic glyphosate resistance? Will you look for SNPs in promoter regions or in other cis-trans regulatory elements?
So, so we haven't found a metabolic resistance specifically for glyphosate resistance. What we might do is for glufosinate resistance. That might be the case because we found enhanced metabolic resistance in the populations that we studied. So the next step would be to find a map, and hopefully we can find a map to that metabolic resistance gene. And then we are going to have to do some, just look more carefully at the specific genes and see if there is any SNPs, deletions, perhaps sequence or transform new plants, with the promoter region. I don't know. I haven't really gone that far yet, but I think the first step is going to be, the next step is going to be find map.
Mm-hmm. I think you might be answering the next question from John Lamas, who says, in each case, you've identified regions of the genome that are very likely to play a role in resistance and gathered a number of genes that may be responsible for this resistance. How do you plan to hone in on specific genes from these regions that are imparting herbicide resistance?
Yeah, I think the next step is going to be again to do the find map because we obtain very large blocks of the genome associated with resistance. So if we can find map, we can narrow down that interval, and hopefully we can get a handful of genes that we can transform rhabdopsis. We can probably do a few, like a 3 or 4 genes transform, and it wouldn't be too hard, and it's doable. The problem is when you try to do 800 genes, it's just not doable. But hopefully we get a few genes that we can work on.
And I think you might have addressed this next question, but specifically, what was your approach to find map the QTL?
Find a map QTL. I think the next step is going to be get a large population. We will probably, and this is still on the works, but we will likely use CASP assays because we already have CASP assay is just a type of PCR or like a single nucleotide polymorphism assay that you run in the qPCR, or like a fluorescence meter or something, and that you can look for specific SNPs in the genome. You can design them, and we can do that because I have the genomic information already. So we can just get a large F2 or F3 population, phenotype it, and use a CASP assay.
All right, one last one before we hit the 1 o'clock mark. You mentioned that you generated Hi-C data. Did that not have any information pertinent to the herbicide resistance you were looking at, or is it solely looked at for the genome construction process?
Yeah, that's a good question. I only use the Hi-C data for assembling the genome, and that's because I only generated Hi-C data from the susceptible individual that was being used for genomic for the genome assembly. Yeah, so only use that for the genome assembly, not for herbicide resistance. No.
You left us with 2 minutes. I'm going to ask you one more. It says, thanks for the great presentation. Did the differentially expressed genes from the RNA sequence analysis overlap with the QTL regions identified? And how did you decide on the 2 megabase and 50 megabase regions?
Yeah, that's a good question. So for the differentially expressed gene in the RNA-seq I had 2 RNA-seq data sets. For paraquat resistance, yes, there was a really good overlap, and so did for glufosinate. So yeah, so one of the genes that was highly expressed for glufosinate was also located in the QTL region. Yes. And how I decided on the 2 megabase, that's totally random. Like it's super subjective how you decide on the 2 megabase. The 50 megabase region, I chose based on the QTL interval. So that's statistically supported, right? So we have the QTL interval. We have the statistics that says, okay, 95% chance that this region from this location to this coordinate, it is associated with resistance. So then I chose the 50 megabase. I think you're referring to 2 megabases, you're referring to the paraquat resistance study where I took the 2 megabases regions around these statistically significant SNPs, and that was a random number that was picked. Yeah, so super. What ideally one could do is to understand patterns of how the genome they are. Well, never mind. I think yeah, I'll leave it there.
All right, we're going to call it there. Thank you, everyone, for coming to this webinar. We'll see you all at the next one in about a month. So look for the announcement for that coming from Kaio and from the IWGC. We appreciate everyone's time. Thank you.
Thank you so much. I appreciate your time and thanks for coming.
Great job, Kyle.
Thanks, Doc.
That was really good.
Glad you made it.
See ya.
Recording now.